Okay, we're going to talk about lipid-soluble messenger molecules, or basically lipid-soluble hormones, what we're talking about, such as cortisol, with the basic structure outlined in here. That is typically something like this. And like this. So why are we talking about this? So we're talking about lipid-soluble hormones such as cortisol, estradiol, thyroid hormone, and calcitriol. They are hydrophobic, and therefore they can diffuse readily across plasma membranes. So what basically this means is that because here we have a nice blood vessel, so you have all the red blood cells going, and you have some of the actual messenger proteins here. Because they are poorly soluble in plasma, what they actually do is they circulate with bound transport proteins, for example, albumin, CBG, TBG, in the blood, and only the unbound free hormone is actually biologically active. So when they are at a target cell, what happens is that these fat soluble or lipid soluble, for example, a hepatocyte responding to cortisol, this free hormone diffuses directly through the phospholipid bilayer, and that come on, give me a drawing. And that's what's happening with all of these lipid soluble hormones. They can directly pass through this lipid phospholayer. Inside the cytoplasm, they go in and they bind into, for example, cortisol binds a glucocorticoid receptor. You have the cortisol over here and it binds into the receptor over here. But it's floating around in the cell. The steroid receptors are typically cytosolic, whereas thyroid hormone receptors are inside the nucleus, but this is outside. So what happens is that then this binding produces a conformational change, which then allows it to actually pass on to the nucleus, which is over here. The nucleus barrier is over here. So then it actually translocates into the nucleus. And in the nucleus, this complex binds to specific DNA sequences called hormone response elements, HRE, in the DNA, controlling and modulating the transcription of the different target genes, for example, and binds in here and enables something to happen. The newly generated mRNA, which is then generated from here, mRNA, which is enabled by this hormone, is then exported to cytoplasm side, outside of the nucleus, and translated into proteins that alter the cellular function. In the cortisol example, this includes the upregulation of glioneogenic enzymes such as PEPs-CK. So, what makes this special? So, because of this, mechanism depends on gene transcription and protein synthesis. Lipid-soluble hormones exhibit actually a slower onset of action because they are slow to get into the cells, and you only have a few of them. But they produce prolonged physiological effects because they can stay in there as well. So that concludes our quick intro to lipid-soluble hormones.